Good morning to all. So, we will continue with remaining foodborne infections. So, in this lecture, we are going to see different rickettsial foodborne infection. So, Q fever is again important foodborne illness, and that is caused by Coxiella burnetii and has an incubation period of 2 to 4 weeks. So, again this is important foodborne infection, and it is mostly caused by consumption of unpasteurized or raw milk, and symptom includes sudden onset of fever, then dry cough and chest pain due to pneumonitis, and hepatic disorders which include slight jaundice occur in severe cases, and mortality is low with complete recovery. So, this is a rickettsial foodborne infection in that Q fever, and it is caused by Coxiella burnetii. Okay. As far as epidemiology is concerned, so Q fever is mostly an occupational disease among people who handle livestock and raw animals products, example farm and slaughterhouse workers, and infection occurs through consumption of raw milk, then contaminated butter as well as cheese, and infection may also occur through contact with infected placentas and contaminated straw beddings and animal carcasses and slaughterhouse offals. So, all these are sources through which this microorganism entered into the main food stream. And in case of diagnosis, so we have to perform serological test, and serology includes CFT, then microagglutination and FAT. So, these are several diagnostic tests for this Q fever to identify this Q fever bacteria, that is Coxiella burnetii, and isolation of this bacteria, it should be carried out in well equipped laboratory due to high risk of infection. And in case of control measures, so pasteurization of milk and milk products. So, pasteurization is the best method to eradicate this pathogen and to avoid this disease. Heating of milk or milk products at 63 degrees Celsius for 30 minutes or 72 degrees Celsius for 15 seconds. So, these are different pasteurization methods that are commonly used for eradication of this pathogen, and safe disposal of offal. So, these are different methods by which we can control Q fever causing foodborne infection. Then foodborne intoxication. So, now what is meant by intoxication? So, intoxication is the toxic shock syndrome which is caused by toxins that are produced by different types of microorganisms after entering into your body, and so this term is known as intoxication, means poisoning due to toxins that are secreted by different groups and different numbers of different types of bacteria when present in food. And so, these are diseases caused by consumption of food containing biotoxicants which are found in tissues of certain plants and animals. Then metabolic products that are better known as toxins formed by and excreted by different types of microorganisms. It includes bacteria, then fungi, then algae, while they multiply in food or in gastrointestinal tract of man. Then poisonous substance which may be intentionally or unintentionally added to food during production, processing or transportation or storage. So, overall these are main sources of foodborne intoxication, in that biotoxicants, then metabolic products, toxins and poisonous substance. These are things responsible for foodborne intoxication. So, foodborne intoxications have short incubation periods, up to minutes to hours. So, these are very rapid in case of their symptoms, and the symptoms may appear or manifest within a few minutes to hours and are characterized by lack of fever. And foodborne intoxications are classified into bacterial intoxications, then fungal intoxications, then chemical intoxications, plant toxicants and poisonous animals. So, oral intoxication includes toxins secreted by bacteria, then toxins secreted by fungi, and then some chemical toxins that are present due to some other sources, then plant derived toxicants and poisonous animals. So overall these are foodborne intoxicants, and this is the classification. Okay, then in that we'll first see bacterial foodborne intoxication. So in that we're going to see Staphylococcus aureus intoxication, then Bacillus cereus foodborne intoxication, then Clostridium perfringens foodborne intoxication, and Clostridium botulinum foodborne intoxications. So these are main bacteria and their toxins that causes intoxications of foods or foodborne illness. Then Staphylococcus aureus foodborne intoxication. So this is a type of foodborne intoxication which is caused by consumption of food contaminated with staphylococcal enterotoxin produced by certain strains of Staphylococcus aureus while growing in food. So this staphylococcal foodborne infection or intoxication, it is mainly caused by Staphylococcus aureus, and this bacteria secretes enterotoxin that is known as staphylococcal enterotoxin, and it affects stomach part of our body. That's why it is categorized under enterotoxin, and this toxin is produced by Staphylococcus aureus while growing into food. So the organism produces following 5 serologically different enterotoxins that are involved in foodborne intoxication, and these 5 enterotoxin includes Staphylococcal enterotoxin A, then Staphylococcal enterotoxin B, Staphylococcal enterotoxin C, Staphylococcal enterotoxin D, and Staphylococcal enterotoxin E. So these are 5 types of enterotoxins that are secreted by different types of Staphylococcus aureus, and all these are enterotoxin. They cause severe effects in our body. So individual strain of Staphylococcus aureus may produce one or more enterotoxin type while growing into food. So these all types of toxin, it may be produced by one type of bacteria or several different types of Staphylococcus aureus bacteria. Then growth conditions. So as far as Staphylococcus aureus is concerned, it is facultative anaerobic bacteria. It does not form any kind of spore, so it is non-spore forming gram positive coccoids. It grows at a range temperature between 12 to 44 degrees Celsius, and optimum growth temperature for this bacteria is 37 degrees Celsius. That is, it is well growth at body temperature, and pH range for this bacteria is 4.0 to 9.83, and optimum pH range is for this bacteria is 7.4 to 7.6. So growth occurs in an environment containing up to 18% sodium chloride and water activity of 0.86 to 0.88 when growing aerobically, and 0.9 under anaerobic conditions. So these are overall information regarding growth conditions of Staphylococcus aureus. Then toxin production. So toxin production occurs at a growth temperature at 12 to 44 degrees Celsius and at pH 4.2 and at salt concentration greater than 10% or equal to 10%, and no toxin production occurs at temperature below 12 degrees Celsius and pH greater than 4.2 and salt concentration less than 10% salt. So these are important parameters as far as toxin production are concerned. So you can see toxin production occurs at optimum growth temperature, that is between 12 to 44 degrees Celsius, and pH range for toxin production is 4.2, and salt concentration needed for toxin production is less than or equal to 10%, and no toxin production occurs at temperature below 12 degrees Celsius, means at freezing temperature toxin production is not likely to be occur, and similarly at pH greater than 4.2 and salt concentration less than 10%, these are the conditions where toxin production get stopped or no toxin production occurs. Then nature of enterotoxins. So all staphylococcal enterotoxins are heat stable. It can withstand heating up to 100 degrees Celsius for 1 hour, and ordinary cooking procedures, pasteurization and drying do not inactivate these enterotoxins. So this is again important information as far as these toxins are concerned. So these are heat stable. These toxins, they are still active after heating at 100 degrees Celsius for 1 hour, and ordinary cooking procedures like boiling, then cooking food on heating, then pasteurization of different foods or milk or milk related products, they still cannot destroy these toxins, and these enterotoxins are still stable after following these procedures. So they are intensive to pH changes. So pH is again important factor for toxin production. So any fluctuations in pH can alter the toxin production rate, and they are insensitive to pH range between pH stable and resistance to most proteolysis enzyme like trypsin, chymotrypsin, renin and pepsin. So enterotoxins are also not affected by irradiation. So all these 5 enterotoxins have similar potency. So all these 5 toxins have similar potency, means these 5 types are able to cause severe infections. Okay. So as far as competition with other organisms, Staphylococcus aureus is a poor competitor and therefore grow poorly or not at all when grow together with other organisms. So majority of Staphylococcus aureus food poisoning are due to foods in which microbial flora is substantially reduced, such as cooked or cured or pasteurized foods. Then vehicles for contamination of these intoxications. So milk and milk products including pasteurized milk, then yogurt, then chocolate milk, then fermented milk and cream filled pastries, then poultry, then fish and shellfish, meat and meat products, non-meat salads, then egg and egg products, then different types of vegetables and cereal products have been involved. So these are all sources through which Staphylococcus aureus may contaminate these foods. Then reservoirs. So staphylococci are found in varying numbers in air, dust, then water, then foods, then fecus and sewage. And the primary habitat of Staphylococcus aureus is the mucous membrane of nasopharynx and skin of man and animals. Then organism is found in nose, then skin and saliva and intestinal content and fecus. So human carriers of these organisms are numerous and are undoubtedly the source of number of outbreaks. Then reservoirs continues with contamination of foods may be tracked to food handlers and minor septic hand infections or severe nasal infections. And the nasal mucous membrane is another particularly important source of staphylococci of human origin. So all these are reservoirs and sources of contamination. So staphylococci are normally present in your body as a part of normal body flora, and it may cause opportunistic infections and it may contaminate through these different body parts. So disease symptoms in man includes incubation period is 1 to 6 hours after consumption of food and contaminated with at least 1 microgram of enterotoxin. Clinical sign includes salivation, then nausea, then vomiting, then abdominal cramps and some diarrhea with prostration. It has attack rate of 5 to 100%, but facilities which occurs in children, the old and debilitated victims are rare, and duration of illness it lasts for 24 to 72 hours, and dose of 1 microgram or more is needed to cause disease. Then diagnosis. So in case of diagnosis, so you can use of clinical symptoms. Then incubation time, it is for 1 to 6 hours. Then clinical symptoms includes nausea, headache, vomiting as well as diarrhea, and enumeration of organisms in food and stool you can use for diagnosis of this particular intoxication. And Enumeration of organism in food and stool, so it may be greater than 10 raised to 6 CFU per gram of Staphylococcus aureus in food, and indicative or involvement of disease in an outbreak. So enterotoxin detection is suspect of food, stool and in vomitus of victims using various methods, which includes serological test, example ELISA, then reverse passive latex agglutination test, or biological, example monkey feeding test, and use of molecular biology techniques, that is gene probing and polymerase chain reaction. So by using these techniques you can diagnose the occurrence of staphylococcal intoxication. Then as far as preventive measures are concerned, so practice of good personal hygiene including good personal conduct in food establishment and when handling of food. So use of spoons when serving foods to prevent contamination of cooked foods, as well as fast cooling of cooked foods and keeping such foods at low temperature. Then discourage consumption of leftovers. The next food intoxication is Bacillus cereus food intoxication, and this is foodborne intoxication caused by consumption of enterotoxin produced by some strains of Bacillus cereus, and the organism produces the following enterotoxin which are involved in foodborne intoxication. So two diarrheal enterotoxins, hemolysin BL enterotoxin and non-hemolytic enterotoxin, then emetic toxin. So these are different types of toxin that are secreted by Bacillus cereus, and this leads to severe intoxications and complications as far as food intoxication is concerned. So Bacillus cereus is commonly found in soil and it is saprophyte, and it is easily spread to many types of foods, especially plant origin, and it is frequently isolated from meat as well as egg and dairy products. Then cereal dishes, example rice, spices, then mashed potatoes, then herbs, then vegetables, then means meat and cream and milk pudding have been involved in Bacillus cereus poisoning. So these are different sources through which these bacteria can enter. These are, or after ingestion of these kind of foods, we can cause intoxication that is caused by Bacillus cereus. Then as far as symptoms of disease is concerned, so Hematic Syndrome, so the syndrome is characterized by nausea, vomiting, then abdominal cramps and sometimes diarrhea that occur for 1 to 6 hours after consumption of contaminated food, and the syndrome is associated with ingestion of rice as well as pasta based foods, and these all syndromes are categorized under Hematic Syndrome. Then diarrhea syndrome. So in case of diarrhea syndrome, patients experience profuse diarrhea, that is watery stool, then abdominal cramps and tenseness, rarely vomiting, and beginning of these symptoms occurs after 8 to 10 hours after ingestion of contaminated food, and fever is absent and symptoms resolve within approximately 12 hours. So this is good thing. So then diagnosis. So diagnosis, you can use clinical signs as well as symptoms for diagnosis of this particular intoxication illness, and enumeration of organism in food using selective media such as Bacillus cereus strains of same serotype should be found presenting significant numbers greater than 10 raised to 5% CFU per gram of microbial load in incriminated foods and in fecus and vomiticus and affected person. So detection of enterotoxin in foods. Enterotoxin detection has been done using biological as well as immunological methods, and in case of prevention, so good hygiene should be observed in food established and by food handlers, and proper cooking of foods to destroy spores, and keeping food at low temperature and fast cooling foods. So again, these are preventive measures. The next intoxication is intoxication that is caused due to Clostridium perfringens, and this is again foodborne intoxication caused by Clostridium group of bacteria. In that, Clostridium perfringens enterotoxin that is abbreviated as CPE, produced in gastrointestinal tract by enterotoxigenic strains of Clostridium perfringens. So these organisms found in soil as well as dust and water and sewage, marine sediments and decaying material and intestinal tract of humans and other animals. So this organism is spore forming anaerobic gram-positive bacilli. So Clostridium perfringens intoxication includes food poisoning. Strains have a variety of origins including humus as well as animal fecus, then abattoirs and sewage as well as flies, and spores produced by these organisms can resist for boiling for 4 or more hours, and in case of these spores are present as a contaminants on raw meat, they may resist boiling or steaming, and on slow cooling the spores will germinate into rapidly multiplying bacterial cells which produces large amount of toxins. Then causes of intoxication. So Clostridium foodborne intoxication is caused by ingestion of food containing large number of vegetable cells of enterotoxigenic Clostridium perfringens type A, and some of the types that are type C and D strains also cause this intoxication illness, and these cells multiply in intensive and sporulate, releasing Clostridium perfringens enterotoxin, and sometimes CPE may be preformed in food and may once the food is consumed, and symptoms may occur within 1 to 2 hours. Then characteristic of CPE. So Clostridium perfringens enterotoxin CPE is synthesized during sporulation, and CPE is heat liable. It gets destroyed at 60 degrees Celsius for 10 minutes, and its activity is enhanced by trypsin. So food poisoning strains are heat resistant and survive heating at 100 degrees Celsius for 1 hour, and vehicle of food. So the food involved are those that are prepared one day and served the next day. So food that have been involved includes red meats, then chickens, then fish, then porks, then fruits, vegetables and spices, etc. The heating of such a food is inadequate to destroy heat resistant endospores, and upon cooling and warming the endospores germinate and grow. So endospores are again heat resistant, and the toxins they are produced during this sporulation phase. So the cooking kills the vegetable cells of Clostridium perfringens but inactivates surviving spores which will germinate again, and they will multiply again. So food poisoning occurs when the level reaches 10⁻⁷ to 10⁸ cells per gram of food, and growth is enhanced by anaerobic conditions achieved after removal of oxygen by cooking. Then mode of transmission of to foods. So these bacteria can directly enter through slaughterhouse or directly from slaughterhouse animals. Then contamination of slaughter meat from containers, handlers, then dust and water, then cross contamination in kitchen environment. So all these are sources or mode of transmission to food. Then symptoms of this disease in man. So symptoms appear within 6 to 24 hours after ingestion of large number of viable vegetative cells, up to 5 into 10⁸ gram of food, but not after ingestion of spores. So symptom includes nausea, then intestinal cramps, then bronchitis, diarrhea, and vomiting is rare, and the illness takes almost 1 to 2 days duration. Then diagnosis. So for diagnosis of the disease you can use clinical signs as well as symptoms. So typical symptoms includes abdominal pain and profuse diarrhea, and it lasts for 12 to 24 hours following consumption of food, and enumeration of Clostridium perfringens in fluid and stool. So you can enumerate by culturing or simply by other methods. Then detection of enterotoxin in food and patient stool using serological methods like ELISA, then RFL-PA. Then prevention. So proper cooking and proper cooking of food and eating freshly prepared foods, then through washing and sanitization of containers, then hygiene handling of cooked foods, then fast cooling of cooked foods, then storing foods in small quantities will enhance cooling, and proper reheating of cold food before consumption, and storage of leftovers or unused foods in freezers. So all these are preventive measures to eradicate this disease. The next again important foodborne intoxication is Clostridium botulinum foodborne intoxication. So Clostridium botulinum foodborne intoxication, that is commonly known as botulinum or botulism, is a type of food poisoning caused by consumption of enterotoxin produced by strains of Clostridium botulinum, and Clostridium botulinum is obligate spore forming anaerobe and it is gram positive bacilli. So the strains are divided into proteolytic and non-proteolytic types according to whether they hydrolyze proteins or not. So the intoxication is caused by botulinal toxins of type A, B, E, F and G, and that are produced by Clostridium botulinum type A, B, E, F and G. While the organism grows in food, the Clostridium botulinum types C and D produces toxin C and D that causes diseases in animals. So the type E strains are non-proteolytic while the rest are proteolytic. So this typical feature we can use for identification of this Clostridium botulinum. And spores of Clostridium botulinum type A can survive at temperature up to 120 degrees Celsius. So as far as growth characteristics are concerned, so proteolytic strains grow at temperature which is range from 10 degree to 50 degrees Celsius, while non-proteolytic grow at 3.3 to 45 degrees Celsius, and optimum growth temperature for this bacteria is 35 to 37 degrees Celsius. That is it is almost grows at our body temperature. So toxin production occurs at temperature range between 25 to 30 degrees Celsius. So both strains grow at minimum pH of 4.5, and proteolytic strains produces an active botulinal toxin, while non-proteolytic strain produces inactive protoxin that requires activation by trypsin. So these are overall growth conditions or growth characteristics. So these toxins are neurotoxins. So basically they affect nervous system in our body, and that are highly toxic, heat liable and inactivated by heating at 80 degrees Celsius for 10 minutes, and unstable at alkaline pH but stable at below 7 pH, and these are resistant to pepsin as well as acidic environment. So these are basic features of this toxin. So basically it is neurotoxin. It affects neurons and nervous system in our body, and the toxin can resist the action of gastric as well as intestinal juices. So botulinous toxin is one of the most lethal poison as far as other are concerned, and the calculated lethal dose for an adult person is 10 microgram per. So types of food implicated. So foods associated with anaerobic conditions such as spoiled or scanned meat or hams and bacons stacked without air access are particularly liable to infective, and homemade fermented foods have been incriminated. So together with smoked, pickled and canned foods that are allowed to stand then eaten without adequate cooking. So these are types of food. Then uncooked fresh foods are safe because they are eaten before the toxin has had time to develop, while if foods are cooked, so toxin is destroyed. So these are different types of foods through which we can get illness by intoxication of this Clostridium botulinum. And a role of preservatives in meat. So nitrates and nitrites are used in canned meat as a preservatives, and the salt reduces chances of growing of Clostridium botulinum and inhibit toxin production. So the danger of botulinum has been the deciding factor for in formulation of food processing techniques, especially in case of canned meat. And as far as mode of transmission are concerned, so contamination of food due to improper handling may contaminate, then insufficient heating of food to destroy spores, and spores present in animal tissue, example meat as well as fish. So these are different modes of transmission for this Clostridium botulinum. And symptom associated with the diseases includes adult botulism. So the period of incubation in man is usually 12 to 72 hours, and symptom includes nausea, then vomiting, then fatigue, then dizziness, then headache, then dryness of skin, then mouth and throat ulcers, then constipation, then lack of fever, then nerve paralysis and great muscular weakness, and double vision, respiratory failure and death. So overall these are symptoms associated with this type of intoxication. And duration of illness, it may last for 1 to 10 days, and mortality is high in case of this particular intoxication, and it constitutes almost 60 to 100% of affected person, and the earlier appearance of symptoms and higher mortality rate. So these are details regarding intoxication, that is adult botulism, and then next is infant botulism. So this occurs in infants less than 1 year of age following ingestion of spores in honey or syrup, and spores germinate in gastrointestinal tract with toxin production, and a high number of spores are found in fecus of infants during acute phase of the disease, and the number of reduces as recovery progress. So symptoms are similar to adult botulism. And diagnosis, you can use clinical symptoms, then you can isolate this particular Clostridium botulinum strains from foods, and demonstration of botulinum toxins in suspected foods, patient serum, vomit and stools using following methods. So we can detect the toxin by some biological methods, example mouse challenge and protection test, then serological methods, example diffusion, electrophoresis and ELISA. So these are different tests we can perform for diagnosis of this particular Clostridium botulinum toxin. And in case of preventive measures, so proper hygienic conditions is important as far as prevention of this disease is concerned. Then ensuring proper manufacturing practices, example ensure proper sterilization as well as preservation of canned meat. Then preserved foods processing rancid and other orders should be rejected. Then proper heating of food before consumption to destroy heat level neurotoxins, and food should be heated up to 80 degrees Celsius and temperature maintained for at least 10 minutes before heating. So pike-free foods are rendered safe if brine is used. Used contains not less than 10% common salt in weaker brines, and microorganisms can continue to multiply. So ensuring fast cooling of food, this will ensure that spores that may be remaining do not germinate in food, and almost care should be taken into manufacture of cans, their transport, then handling and storage, and subsequent use during packing of the product. So this is overall bacterial intoxications. So in next lecture we'll going to see fungal intoxications.